Good morning to us students. Today we're going to continue the final part of Activity 3, the genotype. Chapter 4, Biological Identity and Genotype. So please open your books to page 80. Please find in this slide the correction of the latest homework assigned to you. If you have any questions, ask me privately. Kindly copy the correction on your copybooks. Biological identity and genetic printing. Every individual has a unique genetic identity, which means having a unique DNA, except for identical twins. The DNA fingerprinting that we studied in the previous video, when performed for all the genome, which means covering all the genetic information or all the DNA, can be used for paternity tests and tests for proving whether two individuals are related. By comparing the bands of the gel electrophoresis, which indicates specific regions of the genome of two individuals, one is able to know the relatedness among them. A very good example is given in the next document. In the next document, you can see the gel electrophoresis for 4 pairs of twins. These twins can either be identical twins that look exactly alike or fraternal twins that don't look alike. As you can see, pairs A and D are fraternal twins since they have different bands at different levels. However, pairs B and C are identical twins as they have exactly the same bands at exactly the same levels. Just like having DNA fingerprints that show several bands, indicating that the entire genome was used, we also have gel electrophoresis that can study specifically one gene. Every gene is found on a specific location on a specific chromosome of the cell. And since we have only 2 alleles for every gene in our body, as shown in the next document, you can see that we have a pair of homologous chromosomes, and at a specific location we have one specific gene that is expressing the A allele on one of the chromosomes that can be maternal, for example, and the other chromosome is expressing the allele O, which is, for example, considered a paternal chromosome coming from the father. Therefore, it will show that gel electrophoresis is going to show only 2 bands indicating 2 alleles of a specific gene. If the 2 alleles are shown in the gel electrophoresis, then the genotype can be easily revealed. Rules to memorize in a genotype. You have to keep this in mind. An individual having a recessive phenotype, A for example, small a, must have a known genotype, where the recessive allele is expressed in a homozygous state. The genotype in this case would be AA, small a, small a. Another rule says that an individual having a dominant phenotype, capital A, can have either a homozygous, capital A genotype, or a heterozygous genotype, capital A, small a. Application. Samples taken from Sam and his normal parents for the proteins of APRT enzymes were run on a gel electrophoresis. Document 1 below displays the results of the gel electrophoresis. The first question is determine the affected allele. Let's read the document together really well before answering the question. You can see that we have 2 levels, level allele 1 and level allele 2. For the mother, the gel electrophoresis shows that she has two bands, one at the level of allele 1 and one at the level of allele 2. Same thing for the father. However, Sam expresses a very thick band at the level of allele 1, which indicates that we have both bands overlapping at the level of allele 1. The given says that the parents are normal. However, Sam is suffering from the disease, which means that Sam has an affected phenotype, and therefore the allele for this phenotype are the alleles for this abnormal phenotype are supposed to be carried by Sam. Sam is expressing two bands at the level of allele 1, which means that allele 1 is supposed to be the abnormal allele. The answer: the parents are normal and showing in the results of document 1 in their electrophoregrams that they possess both alleles shown by two bands, one at the level of allele 1 and another at the level of allele 2, which indicates that they possess both alleles, yet they are not expressing them in their phenotype, which means that the affected allele is masked. However, Sam is affected and shows one thick band at the level of allele 1. Therefore, the allele responsible for the disease is allele 1 and the normal allele is allele 2. Second question. Specify the genotypes of the entire family members by assigning symbols for the alleles. Since we already identified which one is the normal allele, which one is the recessive, which one is the abnormal allele, now we can assign the symbols for them. Let capital N be the symbol representing the dominant allele coding for the normal allele. Let small a be the symbol representing the recessive allele coding for the disease. The genotype of the mother and the father is capital A, small a, since both possess two bands each at the level of alleles 1 and 2. However, the genotype of Sam is small a, since he possesses one thick band at the level of the allele 1, so two copies of allele 1. Kindly note that you have a homework for tomorrow, probing the activity questions for activity 3, page 81, and exercise 6, page 86. Please note that you're supposed to copy all the previous slides clearly on your copybook. Thank you.